this is Mrs. Ness-Deden. Today we're going to be reviewing some things about passive transport that you've learned this week and looking at several examples along the way, including a couple lab demonstrations, so that you can practice applying all that vocabulary that we've been talking about this week. We're going to start by reviewing a little bit about cell membrane structure. And to begin with, we actually have a little demonstration. I am going to show you here. I have here a beaker that contains oil and water. The water is the bottom part, and the oil is sitting up here on top. This is going to serve as a model for a cell membrane, and we'll talk about that in a little while. But for now, I just want you to make some observations. I'll stir it up a little bit so you can kind of see what happens. You've already learned a lot about the chemistry of water, so think about that a little bit as you look at this as well. And try and figure out why these two stay so separate, even when you stir them up. You can see they kind of separate right back out. So that oil and water mixture is an example of something that can serve as a model for the cell membrane. You probably noticed that the oil formed this nice thin layer across the top of the water. And even when we mixed them together, the oil and water didn't want to mix very much. Oil is the same type of molecule as the molecules that make up most of the cell membrane. Oil is a type of a lipid. And since it stays separate from water, you can probably figure out that the water, like the tails of the lipids in here, is hydrophobic. And so it separates out from the water and creates that nice thin layer on top. You can actually think of the cell membrane kind of like a little bubble of oil around the outside of a cell. That's very much like what it is. It also has that same property that we talked about with the cell membrane of fluidity. That layer of oil can kind of bend and shift and move as it surrounds the cell. As you look at the diagram on the screen, though, there is one part of a cell membrane that is not in that model. Oil on water does a good job of representing the lipid bilayer part of the cell membrane, but the part that we're missing are the proteins. Right? Maybe to improve our model, we could float some things in the oil to represent those proteins. The cell membrane is really kind of like a layer of oil around the outside of a cell. That's that lipid bilayer with proteins that float in it and through it that serve as protein channels and identification and serve other functions for the cell. Today, we're going to look at how things cross this membrane using passive transport. Remember, passive transport does not require any energy. It's similar to a downhill movement that just happens naturally on its own. And to start with, we're going to set up one demonstration, kind of like the carrot lab. This one needs to run for a while. There needs to be some time for things to cross the membrane. So we're going to set it up now, and then we'll come back to it towards the end of our video. As we set up this demonstration, take a minute while I shift the camera and get things ready to copy down this data table in your notes. As you watch today, you are going to want to take some notes on some of the examples we use. All of the examples and things we talk about today will be really helpful as you work on the passive transport homework that's due on Friday. So take a minute and copy down this data table while I get some things set up for us. She's just setting stuff up in front of a camera is obnoxiously annoying. It's just like 2 inches this way. Can you still see everything? Okay, now we're ready to start our demonstration. So what we're using as our model in this demonstration is we're using an egg. You can see in the camera here, we have 2 eggs. These are actually raw eggs where the shell has been removed. You can actually take the shell off of an egg just by soaking it in vinegar for a few days. Since vinegar is acidic, it will actually dissolve the eggshell right off. And you may or may not know, but underneath the shell of an egg, it has a membrane. So this is not cooked at all. This is still a raw egg. It's kind of squishy, like a little water balloon, but it's surrounded by a membrane, very much the same way that cells are surrounded by a membrane. So we're going to use this as a model of a cell to look at how substances move in and out of cells. You can also see we have 2 beakers here. One of them just contains distilled water. Remember, distilled water is just pure H2O. There's nothing else in it. And we have salt water, which is as saturated as we can get it, which means there is as much salt in there as would possibly dissolve. Before we put each of these into the solution, we are going to put the egg on a balance and see what its mass is to begin with. So I'm going to zero that out. And we'll start with our egg that goes in distilled water. So this egg right now has a mass of 70.6. So in that box in your data table, write 70.6 grams for the before mass of the egg that we're putting in distilled water. So I'm just going to put that right into that beaker. Our second egg, we'll take the mass of this one as well. A little bit bigger egg here. The saltwater one has a starting mass of 85.0 grams. So we'll take that one and set that in the salt water. You can already notice a little difference. You might notice that the one in salt water is floating. That really has nothing to do with passive transport. That just has to do with the density of the salt water itself, but it's kind of cool and interesting. So we're going to look a little bit at this in terms of the environments that these are in and use a little bit of vocab to explain this. And then you're going to predict what you think will happen. We're going to leave these sitting in here for, I don't know, an hour or two. And then we'll come back and see if and how the mass has changed. So now let's think a little bit about that setup and make some predictions of what you think is going to happen. You should have recorded the starting masses in your data table. You'll see me writing on the screen. That's the writing that'll appear on your screen too. But we're going to draw some pictures and use some vocabulary to describe the environments that we just put those eggs in. So the one in distilled water, we can draw our beaker here. We've got water and we've got the egg. I would suggest that you draw these diagrams in there. A lot of the questions that you're asked on your homework and that you'll be asked on assessments about passive transport have to do with diagrams. So the more you practice with pictures, hopefully the more sense this will make. So when we think about this one, the water surrounding the egg is just H2O. There's nothing else in there. But inside the egg, there's some water. The egg is becoming a living thing, and so it contains mostly water, but it has other things in there too, right? There's some proteins, there's some sugars, there's some other things like that that are dissolved in the egg. So if you look at this, compared to the inside of the egg, we can label what type of solution this is using the vocabulary that we learned yesterday, or a couple days ago. Because the concentration in water, because the concentration of salt and dissolved substances in the water is lower, there's none, than inside the egg, we would call this a hypotonic solution. The environment around that egg is hypotonic. It has a lower concentration compared to the inside of the egg. The inside of the egg would be hypertonic in comparison to that beaker. The other one that we have, the saltwater setup, has water and it's got the egg, and the egg still has a little bit of stuff dissolved in there. But remember, we put as much salt into that water as we possibly could. So if we did that right, our salt water should be very, very highly concentrated. You should have lots and lots and lots of salt in that water. So this environment, compared to the inside of the egg, has a higher concentration. So we would call this a hypertonic environment. Take a minute and think about what you learned yesterday and what you saw in the carrot lab a couple days ago. Sorry, what you learned on Tuesday and what you saw in the carrot lab on Tuesday. And make a prediction. What do you think is going to happen to the mass of each egg? And see if you can explain why. We're going to come back to this demonstration in a couple hours and see how much the mass has changed and see whether your predictions were correct and try and explain it based on what we know. So while our eggs are sitting in their solutions, we're going to look at a series of diagram-based problems to kind of review how this process works and use some of the vocabulary to explain why particles will cross a membrane. So to begin with, we're just going to review a couple of the main words that we use to describe solutions. And we already used these once with the eggs. When we refer to the concentration of a solution, in other words, how much solute, how many dissolved particles there are within a certain volume of liquid, or you can think of it as how packed together those dissolved particles are, we can describe those concentrations as hypertonic, hypotonic, and isotonic. These are three words that we use a lot when we talk about passive transport. Hypertonic, the way that I remember this is, you know that when you are hyper, you have more energy than usual, right? So a hypertonic solution has more solute dissolved in it than on the other side of the membrane. So a hypertonic solution, if this is my cell membrane right here, one side of the membrane might have a lot of dissolved particles on it, and the other side might only have a few. The side with a higher concentration of dissolved particles would be the hypertonic side. In this case, it's the side that I drew to the left of my cell membrane. The opposite of that word is hypotonic. which means it has a lower concentration of solutes dissolved in that solution. And so that would be the right side of the membrane that I drew here. Hypertonic and hypotonic are words like more or less. They're just describing relative to the other side of the membrane, which side has more dissolved particles per volume, per unit volume, and which side has less. When there's a difference like the picture up here, where one side is higher and one side is lower, we call that difference a concentration gradient. That's just a fancy way to say there's a difference in concentration, and because of that difference, the particles are going to start to move. And with passive transport, they always move downhill, in other words, from high concentration to low concentration. So these particles, assuming that they're able to cross this membrane, would start naturally moving from left to right. And they're going to move that way until the concentration gradient, until that difference in concentration is gone. That won't be when the particles all move to the right, because then they're still different. This will stop when they reach an isotonic state, where they have the same concentration on both sides. So that would look kind of like this. That's our cell membrane. That's probably not the same number of particles, but you get the idea. This is an isotonic state. We can also call this equilibrium. In reality, these particles aren't totally going to stop moving. Occasionally, one of them might kind of float through the membrane this way, and one of them might float back through this way. They're still moving, but overall, the concentrations are staying equal on both sides of that membrane. That's an isotonic state, where the concentrations are equal. Now we're going to look at a series of 4 or 5 diagrams, label them using this vocabulary, and talk about what types of movement would occur. So here's the first one. This one is mostly just kind of a review of some of this vocab. You can see there are a couple of things that are already labeled on here for you. So the first piece of advice, anytime you see a diagram, make sure you read all the labels to see if you have all the information. So on the left, we have a picture of kind of a U-shaped tube with a semi-permeable membrane right there. And you can see they've kind of given you a blown up version of that picture here, where they show the semi or selectively permeable membrane running right through the middle there. And you can see the molecules a little bit more closely. On the left side, we have a few of these green particles. That could be sugar or salt. Oh, it tells us up here that this one is showing us sugar. A few on the left and more on the right. So we can put our vocab words in here. We just said that a lower concentration is what we would call hypotonic, and a higher concentration is what we would call hypertonic. And so what they're showing here is the process of osmosis. You see that word down here. And it shows and tells us here that water is moving from the left to the right, which if you look at that, it should make sense. When you look at this kind of blown up picture of this, there's a lot of water molecules on the left. There's a high concentration of water on the left and much fewer on the right. And so overall, those water molecules are going to be moving from left to right. And so at the end, you get this kind of weird thing where the side on the right actually has more water than that on the left. This is still considered isotonic, and this is a really important thing to remember. The word isotonic means they have equal concentration, not equal amounts. And concentration is just the number of particles per unit volume. So let's see if I can kind of illustrate this a little bit. So if we draw a box in here, Just say we make a box about that big. We'll see how well this works. Right now, in that much volume of liquid, there are 4 of those green dots. If we move it over here, we've still got about 4 dots in there. If we move it over here, we've still got about 4 dots in there. So these dots are evenly spread out. You have the same number per unit volume. So this is isotonic. Even though they're different volumes, the concentration is still equal. So we're going to take some of those ideas and start applying them in this next problem. I've been using the word solute today. The solute is the particle that's dissolved into the water. And so you also need to pay attention. In this diagram, it's a little bit tricky to see the difference between the solute molecules and the water molecules. It tells you the solute is the slightly larger one. So I'll outline those in red just so they're easier to see. I think that's a solute too. And again, we have a semi-permeable membrane in the middle. And this question asks us which direction the water is going to move. So first, if we use our vocabulary to label these, which side would you call hypertonic and which side would you call hypotonic? Remember, those words are looking at how much of the solute is dissolved into the solution per unit volume. So kind of how packed together those particles are. There's a higher concentration of solute on the right. So this is the side we would label hypertonic. Ran out of room for that word. And the side on the left we would call hypotonic. But the question here asks us about the movement of water. So you do need to read the questions carefully. A lot of times the solute, in this case these big particles, can't get through this membrane. Only the water can. And if you look at the water particles, there's a lot more water on the left than there is on the right. So here we have a higher water concentration and a lower water concentration here. Water moves through osmosis. And remember, osmosis is just a specific type of diffusion. And diffusion is always movement from high concentration to low. So water is going to flow down its concentration gradient like any other particle. So it's going to move from where there's high concentration to low concentration. And the water would move to the right. Just a couple more examples here to take a look at and think about. Again, this one asks us to label the solutions as hyper and hypotonic and label which way the water will move. So this is very similar to what we just did. This time, if you read it, tells us the sugar molecules are these larger ones. So again, I'll just kind of highlight those so it's easier to tell the difference. And we have a semi-permeable membrane in the middle. So take a minute, label the hyper and hypotonic sides here, and draw which way you think water would move. Actually, copy this diagram into your notes. If you haven't been writing any down yet, you should have a couple of these in your notes as examples. So copy this one down. It's pretty simple. Label hyper and hypotonic and which way the water would move. Did you get that one right? The left is hypertonic because there's a higher concentration of sugar dissolved in it. The right is hypotonic, but the water is going to move from where there's a lot of water molecules to where there are fewer water molecules. So water will move through osmosis to the left side of this diagram. Again, some practice using that vocabulary. Take a minute and look at this diagram, identify which side is hypertonic, which side is hypotonic, and explain what's happening here. This shows you, this is kind of the picture at the start, and this is the picture at the end. Name this process and explain why this happened. How'd you do on that one? Take a look at what I wrote up here for the answers. If you have a lot of questions about this still, make sure you check in with your teacher. Continue to go back and review the notes that we have and ask questions. This can be a little bit tricky, but the more you practice, the more you will be able to interpret these diagrams and figure out what's going on. The last one we have for today looks an awful lot like the egg situation that we started at the beginning of this video. One note about this beaker over here should really have lots and lots and lots of black particles over in here, all the way through the whole thing. It just took a really long time to put that together, so you just have to imagine that there's black dots all the way over there too, okay? So, here we have cell A and cell B in different types of solutions. Just like our eggs, these solutions are different with regards to the cell that's in them. And these two cells aren't eggs, they're actually cells. And you can see they're a little bit different. But cell A has a few particles inside of it, but there are lots of particles outside. So this one is in a hypertonic environment, or a hypertonic solution. And you would call the inside of the cell hypotonic. And you can see in this one that the arrows are pointing out. This one's a tiny bit trickier to understand because it doesn't draw the water molecules for you. But remember, in between the solute particles, the rest of that is water. And so inside of the cell, all of this space is full of water. The cell is a very, very high percentage of water. And out here, because there's so much solute dissolved in there, there's actually a lower percentage of water in between all these particles. And so water is still going to move from where there's a higher concentration of water inside the cell to where there's a lower concentration outside of the cell through osmosis. Cell B has several particles dissolved inside the cell, but only a few out here in the solution. And so this would be considered a hypotonic environment or solution. And the inside of the cell is now hypertonic. So in this case, the water would move into the cell. There's a lot of water out in the solution and a smaller proportion of water inside the cell. So here, our arrows would point in to show that water is moving in. We're not going to answer question F there. It says draw a situation where water would move into a cell. We're going to skip that for now because that's actually very close to what we're going to be doing with our eggs in just a minute. So let's check on those eggs and see how they're doing. So here we are back with our egg demonstration. These have been in this solution probably for about an hour and a half, almost 2 hours now, sitting in either the plain distilled water or the salt water. So we'll go through and we'll take the final masses of these as they come out of here and kind of see what happened. As I take these out, I'm going to dry them off just a little bit so we're not taking the mass of the extra water on the outside. Think back to the predictions you made at the beginning and kind of what you expect to see happening in here. You can also just take a look and make some observations about these eggs right at the beginning. Do you notice any differences between the one that was in the distilled water, that's this one, and the one that was in the salt water? I'm going to take the mass of each of these now to see if that has changed. So our distilled water started at 70.6 grams. It now has a mass of 79 grams exactly. So write that down where you were recording your data for this demonstration. The egg that was in the salt water here, you can take a look at that, started with a mass of 85 grams, and it is down to 75.6. While I switch the camera back to the data table and the screen so we can talk about this, take a minute and see if you can explain these results based on the examples that we just worked through and using some of the vocabulary that you've learned in the past couple days. So now we're back to the data table that we started with. You can see we have our starting and ending masses of these. And we had drawn before a picture of kind of the environments that these were in. And I forgot to label the saltwater one. This one was in a hypertonic environment. And so as you can see, it's pretty clear here that the one that was in distilled water gained mass. So something was moving into that cell, or into that egg, I should say, which is a model of the cell. And if you think about it, there's only one thing that could possibly be moving in that direction, because the only thing in that beaker is plain old H2O, just distilled water. So this process, this has to be an example of osmosis. Remember, osmosis is the diffusion of water. So since the solution in the beaker was 100% water, and inside the egg was less than 100% water, like most living things, eggs are maybe, 70% water. Water moved from where there was a higher concentration to where there was a lower concentration. It moved into that egg, and the egg increased in mass. The one in the salt water lost quite a bit of mass. And if we left it there longer, it would probably lose even more and kind of start getting all shriveled up. And so we can draw our arrows here pointing out of our egg. In a hypertonic environment, water will move out of a cell because there's a higher percentage or concentration of water inside the cell compared to in the solution surrounding it. You know that it's the water moving, not the salt, because if the salt was going to diffuse across this membrane, it would go from high to low concentration. So it would go from outside to inside, and this egg would gain mass, but that didn't happen. So it has to be the water moving out of the egg that caused this egg in the salt water to lose mass. This can happen with real live things. This is why plants don't do very well if they are watered with salt water. This is part of what makes your skin feel so weird and your hair feel so weird after you've been swimming in the ocean. Salt water causes water to move out of a cell through this process of osmosis. Hopefully today's demonstrations and practice problems were helpful to you in kind of clarifying all that vocabulary we learned earlier this week and helping to put it all together to understand this process of passive transport. Have a great rest of your day, and we'll see you tomorrow.